Well, next up we have April Goebl from the Denver Botanic Gardens, and she will be speaking about the genetic variation and local climate adaptation in grassland restoration species and the implications for seed sourcing. So April, go ahead and share your screen. Get started.
Okay, let's see. Let me just put this in presenter mode and let me know if you are all seeing the screen you should be seeing.
We are seeing your notes screen.
Oh, okie dokie. Let's see if I can do a switch of that. Hmm. Okay. So, is that look any different?
Yeah, that looks great.
Okay, excellent. Great. Well, thank you for the patience. Thank you for to the organizers for putting together this great conference and community, and for the opportunity to speak today. I've really enjoyed the talks thus far, yesterday and today. So thank you to the other speakers and the participants. It's been really, I think, an engaged group of people and just a lot of fun so far. I am participating from Colorado in the United States. I am on ancestral, unceded, and current land of many indigenous people and nations, including those of the Arapaho, Cheyenne, and Ute tribes. Today, I want to talk about some research that I'm doing at the Denver Botanic Gardens, thinking about genetic variation and local adaptation to climate and the implications this has for seed sourcing. All right, so I'd like to start with this schematic from the U.S. Bureau of Land Management and the National Seed Strategy, looking at the native seed development process and the different steps from seed collection through propagation and storage and right around to restoration. And the research that we do fits into this evaluation and development step. And we're interested in these questions, thinking about seed sourcing and what it means to identify genetically and ecologically appropriate seed for restoration or plant materials more generally. So we know that evolution causes intraspecies diversity, and this means that not all seed will be appropriate for restoration in a given area. I'm showing this schematic here of a map and a plant that might vary phenotypically across a hypothetical range. And this phenotypic and genetic variation is due to both neutral and non-neutral evolutionary forces. Some neutral factor includes things like natural selection that can lead to local adaptation. And I show these two pictures here just to emphasize the stark differences that we can see in habitats. Plants live in such a variety of locations and experience very different selective pressures depending on their habitat types. And so the fact that there is this natural genetic variation represents both an advantage for restoration, but also presents challenges. So on the one hand, natural genetic diversity in traits is so important in a restoration setting for populations to be able to persist and adapt. And the Bureau of Land Management's Seeds of Success program is doing an amazing job making tens of thousands of collections from wild populations of thousands of species across the United States and banking this genetic diversity and making it in part available for restorations. But the flip side of the same coin is that this natural genetic variation means that not all populations are equal. They may differ in evolutionary history, in ploidy, in fitness at a given in a given environment. And so that means that sourcing, seed sourcing can be challenging. And I think that's illustrated well by the provisional seed zone map, where each color here, this is probably familiar to a lot of folks, each color represents a different climate-based seed zone, and each of the black lines delineates a different ecoregion. And in absence of species-specific information or maps, the recommendation is often to keep seed within a colored zone and ecoregion. And so it's very valuable to have this guidance because species-specific information is often not available. But for this map to be useful across so many different species types, it's going to be making assumptions and largely the one of local adaptation. And so I think it's important to sort of unpack the different components of local adaptation, and to remember that it consists of both the selective pressures driving adaptation or the components of the environment, whether that's climate or other aspects, and then also the traits that are under selection and become adaptive in a particular space, or a particular place in space and time. And so making these assumptions across all or many restoration species in such a wide geographic area is not a trivial thing to do. So that just sort of sets the stage for the research questions that we're asking and why I think they are important. First, we're interested in addressing for individual species if we can identify what traits or proxies for traits or vital rates or life stages are most important for fitness and thus long-term restoration success. So I show this schematic here of a life cycle of a plant, showing that there's different components of fitness and underlying traits to these fitness components at different life stages. And that depending on the species and the environment, there's different contributions of these different life stages and fitness components to overall population fitness and population growth rates. Then we're interested in asking if these critical traits are locally adapted. We may see that that's not the case in some species, or if they are, if we do see signals of local adaptation, what are they locally adapted to? Is it aspects of climate or perhaps other aspects of the environment, including biotic elements? And then we hope then by answering these questions, it will help to improve seed sourcing for individual species by understanding when it makes sense to match by climate or other environmental characteristics, or perhaps mix to increase genetic diversity, to hedge against uncertainty and bet hedge essentially. Or perhaps I use the term maximized here just for the to sort of continue the alliteration. But what I mean is perhaps some species we see that they're optimal genotypes that have ideal traits and ample diversity and perform well across a large geographic area. And so having more species-specific information could help alleviate some of the seed sourcing constraints if the definition of appropriate seed could be expanded in some cases. And this could make a big difference, as we know that procuring ample appropriate seed is a limitation, particularly for large-scale restoration. I'm smiling there and remembering Peggy's talk this morning and the pie chart of just the estimated amount of seed that would be needed to sort of meet some of the goals, and that's pretty huge. All right, so our approach to address this research framework in part involves a set of multi-year common garden experiments in the field using 4 native grassland species. We're focusing on grassland habitats. Common garden experiments can be used to quantify genetic variation by controlling for environment, and also can be used to detect signals of local adaptation by correlating traits to source environment. And so our experiments are being conducted in Littleton. It's south of Denver in Colorado. It's on the Chatfield Farms locations of the Denver Botanic Gardens. This is an aerial map showing a schematic of the layout. The 4 species that we are working with are Artemisia frigida, or fringed sage, Ericameria nauseosa, or rubber rabbit brush, Penstemon virens is front range beard tongue, and Bouteloua gracilis, blue grama. These are common species in grasslands in Colorado and other sort of Western states. Well, the Penstemon actually has a more restricted range. And so we are working with Seeds of Success seed. We are able to obtain some seeds seeds from the Seeds of Success collections to use in this research. So we don't always have a huge number. I'm calling them populations here, but meaning seed source or collections. But we did have high just sample size and within population replication in general. And the data that we've been collecting over the past 2 years and will continue to do so for the next field season and potentially longer include flowering phenology or timing, plant survival, plant size and growth, reproductive output, and leaf traits. So we started by growing plants in the greenhouse, or we germinated seeds in the greenhouse and transplanted our little seedlings to cones or plugs, and then transplanted these to the field. And since we sort of started at this seedling stage in the field, this approach worked quite well. I'll show some data on survival and things like that. But since we weren't able with this approach to sort of get at these important early life history stages like seed germination and seedling emergence, we're setting up a sort of complementary field experiment to look at these life stages in these same species and populations in separate plots. All right, so I'm going to show some results. Starting with Bouteloua gracilis. So a lot of this work is still in development and in the early stages, but I wanted to share some updates on our progress and some of the early results that we are seeing. A lot of the data for this species was collected, and also for the rabbit brush was collected by a graduate student, Megan Clark, who used some of it in her master's thesis. This species, blue grama, is a dominant species in grasslands and is an important species in grassland restorations. So I'm going to be showing results just for 3 of the species today, just to give you a bit of a heads up. So starting here, the collections were from 21 different populations shown on the map here. They are colored by seed zone. There's 9 different seed zones represented. The map in the bottom is just the distribution of the species in North America. And so we saw high survival and flowering rates. So here I'm showing plots of survival rate and flowering rate on the y-axis. On the x-axis is the population. They're ordered by seed zone, as is shown in the legend here. I've circled that medium green seed zone is the seed zone where the Common Garden experiment site is located. And so the points here represent the mean trait value and standard error for each of the populations. And yeah, so we did see pretty high survival. This is after the second year. And interestingly, most of the mortality was after the first growing season, or I thought that was interesting anyways, either over winter or in the second growing season of established plants. For this species, we had the majority of plants flower in the first season, or in the first year, and again in the second year, flowering rates are quite high as well for most populations, above 80%. So then looking at some of the more continuous or sort of discrete numerical traits we looked at, I'm just showing a few here. Reproductive output, or we counted the number of inflorescences. And that's shown in the top box plot here, and in the bottom plot is flowering phenology, or the number of days from planting to the first appearance of the first flower. The boxes are ordered by median trait value. And so we do see that there are, not all populations are different from one another, but in general, there is a fair bit of variation in these traits across the populations. But in addition to comparing variation and averages between the different populations, we're interested in how much variation we also see in these traits within populations, and because that can reflect evolutionary potential and is certainly important in a restoration context. And so to look at that, we calculated the coefficient of variation for all traits for each of the populations. And so that's plotted in this bar graph here. And although summing coefficient of variations might not be biologically meaningful, this is just a visual representation to show the amount of variation in each population across these 4 traits. So I'm showing plant height, growth rate, flowering phenology, and reproductive output. And so, there is some differences in the amount of overall variation that the populations show. Interestingly, I thought, is that the different traits seem to have the same order of the amount of variation across all the populations. And particularly of note, I think, is how phenology seems to be the least variable trait across the populations. And I'll sort of circle back to that, why I think that's sort of interesting to point out here. So then, getting back to the sort of comparisons across populations. So we do see that not all populations perform the same in these different characteristics. And we're interested to know, could that in part be driven by natural selection and local adaptation to climate? So to look at that question, we performed regression analyses using bioclimatic variables from the World CLIM database as predictor variables. It might be familiar to a lot of people, but there are 19 bioclimatic variables that have been generated based on monthly temperature and rainfall data, and they're sort of designed to be more biologically meaningful climate variables and are often used in ecological modeling. And so we used principal component analysis to reduce the number of variables to ones that were meaningful for our populations. For Botulua gracilis, we selected 5 variables along orthogonal axes that had high loadings on the first 2 principal components. So an example of what a regression model might look like using these CLIM variables. So say we were looking at the relationship between the number of inflorescences or reproductive output as a function of these 5 different climate variables. And so I'm just selecting a few results to show here from regression analyses where we're looking at the relationship between trait and climate. Okay, I see I have 5 minutes, so I might speed up a little bit. Some of the sort of main results or sort of stronger patterns that are jumping out here for both flowering amount and flowering phenology, and how these correlate with different aspects of seasonal temperature. The phenology has a relationship with mean temperature of the coldest quarter, or maybe minimum winter temperature. This could be, or I suspect this is quite related to growing season length, which sort of makes sense why that would be tied to flowering phenology, where we see plants from places with warmer winters and likely longer growing seasons flowered later in the common garden. Okay. Oh, the red lines here represent the climate of the common garden site. Okay, I'm happy to talk about some of these results more later if I'm going through some of them quickly. Okay, so next I wanted to show some of the results from Ericameria nauseosa. This is a perennial shrub that's common in Western North America. It's an important forage and cover species for wildlife and food and habitat for pollinators, particularly in the late season. For this species, we had seed from 20 different SOS collections shown here on the map. Again, they're colored by seed zone. We have, let's see, so again, I'm showing results for survival and flowering rate, where the circled seed zone there is the seed zone for the common garden. Again, survival and flowering were quite high, not quite as high, particularly survival as for blue grama, but still relatively high for most populations. I'm sort of looking here now at the probability of flowering, and I sort of found it interesting that plants that didn't flower sometimes, these were ones that, seemed to be struggling to survive, but other times they were big, healthy plants that I think just waited too long because they are used to perhaps having a longer growing season. But then when cold temperatures hit around October at the common garden site, they sort of lost their opportunity. And I think for species like rabbitbrush that's known to be late season flowering, it's, it may be particularly susceptible to sort of climate mismatches when moving seed around for restoration. So now I'm showing box plots for plant size and again, flowering phenology. And for plant size, there seems to be a decent bit of variation across the populations. The flowering phenology, it's a bit harder to tell with sort of the box plot here. And that's in part due to just sort of the resolution we had with the sort of frequency we were doing phenology surveys. But I'll show in a later slide that there is a decent bit of variation that is explained by climate. Again, I was interested in looking at the within population variation. And for this species as well, we see that flowering phenology has lower has the lowest amount of within population variation compared to these other two traits at any rate. And then again, looking at how the between population variation might correlate with climate using these bioclimatic variables. Some of the patterns that jump out are relationships with height and flowering phenology. We see that smaller plants, just on average here, flower later, and they're from warmer and drier places. And then the last species I wanted to present some results for is Artemisia frigida. This is a perennial subshrub that can be drought tolerant and grazing tolerant, and can be important for insects and other wildlife for food and habitat in grasslands. For this species, we had 11 different seeds of success collections. In this map and in the results, each of the populations or collections is colored differently, so they're not colored by seed zone. But there are 6 different seed zones represented. So we don't have as much phenotype data to share for this yet, but we do have a little bit, and we do have some population genetic structure results from genetic sequence data, and graduate student Allison Emery is leading the analysis of this data. So on the right here is a principal component analysis using SNP or single nucleotide polymorphism data, molecular markers, where each point represents an individual and they're colored by population. And so we do see some separation along PC2 that sort of aligns with latitude. And then interesting along PC1, there's separation of this Utah population. The other data I'm showing here, the box plot on the left is of plant size. And here I flipped that on its side just to sort of show how these different clusters of plant size, which sometimes seems to correlate also with growth form in this species and how that sort of maps onto geographic space. All right, so to wrap up here, we saw similar results for blue grama, the Bouteloua gracilis, and the rubber rabbit brush, where we see trait variation between populations on average. Flowering phenology, I think, was one of the really neat traits to highlight, where there was a lot of variation between populations, but notably lower within population variation. This trait is also one that seemed to correlate quite strongly with minimum winter temperature or perhaps growing season. And I think that could suggest sort of an interesting implication for seed sourcing, where it's probably a trait that is important to climate match and optimize for current conditions, but in doing so, we may be having a trade-off where that's not so beneficial for increasing variation or evolutionary potential for future conditions or even current unpredictable conditions. And then for fringe sage, we showed variation in plant size and molecular markers that sort of mapped onto the geographic space. Okay, so quickly to just talk about a few of the next steps, or to continue to sort of integrate some other traits, including leaf traits and data from earlier life stages, and to see just the importance of these for these species, combining other data from or genetic sequence data from some of the other species from common garden plants, and linking that to source climate to see if that also shows signals of local adaptation that might be complementary to the sort of correlative analyses we're looking at with the phenotypes. And then perhaps testing some of these local adaptation hypotheses in controlled greenhouse experiments, these local adaptation hypotheses that we're generating from the correlations with traits or genotypes to source climate. And I want to acknowledge the BLM for providing funding for a lot of this research, which has been really incredible. The Denver Botanic Gardens, which is where I am currently situated, and a lot of folks that have helped in this research. All right, thank you.
Thank you, April, very much for that presentation. We'll open up questions, just a couple of questions so we don't run into the break too long. First question, what's the overlap between seed zones and ecoregions?
I'm not sure totally understand the question with overlap. The ecoregions are I think these were level 2 homework, or sorry, level 3 based on that map, and they're based on ecology, geography, different sort of components of the environment, I think is how those define. There is, a good definition for that I'm actually not super familiar with. The seed zones are based on climate variables, specifically minimum winter temperature and an aridity index. So you can see that there's, or looking at the seed zone map, that the seed zone sort of, those colors can appear in many different sort of geographic areas, but aren't necessarily restricted within a given ecoregion. don't know if that helped at all, but there are probably better definitions out there that I can help provide.
Thank you. And I'll just ask one more. Are there any considerations for selection in the germination and early seedling phase that could happen in the greenhouse before plants get sent out in the field?
Yeah, great question. I mean, it's possible. We germinated seeds, what we say in sort of like optimal, like sort of cushy conditions. The germination and emergence was very high in the greenhouse, and then we randomly selected individuals to be transplanted out into the field. So I think we probably had a pretty good representation, but sure, there always there's possibility of an inadvertent selection.
Great, thank you. I'll pass it off to Morgan.
Yeah, thanks so much, April. It was really cool. Exciting to see about those SOS collections. And now we are going to go into our break. And I think we will reconvene for the next lightning round at 12 p.m. Pacific time. So have a good break. Thanks.